The second talk of this afternoon is from Muhammad Farooq from the University of Calgary, and his presentation is entitled Study of Association Between Drinking Water Quality and Infectious Bronchitis Virus Vaccine Response in Alberta Layer Farms.
Good afternoon, everyone. Thank you very much, Canadian Poultry Research Forum and all the organizers. My name is Muhammad Farooq. I'm first year PhD student at the Faculty of Vet Med in University of Calgary. And I would like to talk about my research project for my PhD that I will be doing in the upcoming 2 to 3 years. And it is about, the water quality in Alberta egg layer farms and its impact and association with the vaccine response. So there is just a little background about the Alberta egg industry. There are 170 registered poultry farmers, the layer farmers I'm talking about. And this is the data of 2021. There were 2.8 million egg producing hens, and on an average the flock size was around 16,500. The total egg production in dozens in 2021 was 77 million. And you know that the system of egg production or farm production, it works on a quota basis. So Alberta has 10.18% of quota from the national stock, like from the national egg production. 10.18 is produced by Alberta in 2021. The egg price of production per dozen was 2.5. And another interesting fact, who do you call a layer farmer? The one who has more than 300 birds is called layer farmer, and he must register himself with egg farmers of Alberta. So a little background about my research. Water consumption is twice as much as the feed consumption. So water is the largest commodity moving inside the barn, but that water comes from different type of sources, maybe groundwater or surface water, and the water that is moving in is not usually treated. So there is a concern. The concern is the water may contain different type of ions like nitrate, sulfate, magnesium, sodium chloride, and they may be contaminated with microorganisms that can come from sewage and animal waste and dead animals. And each of these ions and microbes has direct and indirect consequences for the bird's health. For instance, nitrate and sulfate cause decreased production and less growth in the birds. And sodium and magnesium can lead to laxative effect, wet litters, and the other complications. And the microbial contamination, they cause direct infection of the bird. So there are specific levels for each ions accepted, mentioned in the livestock water quality, a field guide for cattle, horse, poultry, and swine. And we have a background research for the water quality contamination, the one conducted by Sylvia, Susan, and their group. They conducted research on 97 well water. They took samples. They found 20 wells with total coliform positive, 3 with E. coli positive, and 2 with enterococcus. And they also found that only 21% of the well were being tested annually for the chemicals. And another study, which was very large study conducted in Ontario, they found 17 to 24% of E. coli positive well. And this is a very significant study conducted from University of Alberta. They found that the birds exposed to raw water and tap water experimentally only for 7 days, and then they found the raw water has immunosuppressive effect. It is increase the residence macrophages and decrease the CD4 lymphocyte in the abdominal cavity. So the water quality has effect on the immune system. And we know that poultry industry heavily relies on vaccination. In Alberta Layer, only 12 type of different combination of vaccination is being performed every time against the Merrick disease, infectious bronchitis, IBD, Newcastle, and encephalomyelitis. And the other problem is we have a lot of outbreaks, like a study conducted from the University of Calgary, we face a lot of outbreaks of IB, the shellless egg syndrome, the outbreak in 2017. Instead of vaccination, still there are, and it's caused by a mass type of virus, and the vaccine also contains the mass type of variant, but still we get outbreaks. Therefore, we hypothesize that there is an association between drinking water quality and efficacy of current vaccination program implied by the table egg layer industry. Based on the hypothesis, we have two objectives. One is to investigate the association of quality of drinking water and procedure and the efficacy of current vaccination program as determined by serological responses in the table egg layer industry. And then we also have to study the effects of drinking water quality on the efficacy of infectious bronchitis vaccine of layer chicken. We want to study both the humoral immune response as well as cell-mediated immune response. So the first subject will contain questionnaire. So this project is similar like the presented in the previous one, but this is on the air quality. That one was this one is on the water quality. That one was on the air quality. We will be having a questionnaire. We'll get information from the poultry farmers about the barn, its size, dimension, history, and about the birds, its age, production, breed, its vaccination status, about the biosecurity of the farm, about the feed consumption and feed provision, and finally about the disposal of the dead birds. We will get this information through questionnaire, and then we will testing the quality of water, its physical, chemical, and microbiological characteristics. Some tests will be performed on site like pH, total solid, total dissolved oxygen, turbidity, and some will be performed in the lab like for various chemicals and microbiological contaminations. And we will also be taking serum sample from those farms. There are 170 egg producer. We will select 30 flocks, and from those 30 flocks, from each flock, we will select 25 birds for serum samplings at different ages and time points. And we will perform ELISA against IBV, IBD, like infectious bronchitis, infectious bursal disease, and ND, and we will analyze the antibody titers of these three vaccines, and we will correlate this data with the water quality one, how it impacts the titers. So we expect that there would be a good quality water, a medium quality, and a poor quality, but that poor quality water or that less antibodies one doesn't mean it is only due to the poor quality water. So there may be confounding factors, immunosuppressive condition involved, litter condition, underlying diseases, or air quality. Therefore, we will have to conduct controlled in vivo studies to attribute the effectiveness of vaccine of layers to drinking water quality. For that objective, we will do a control experiment, take a day-old chickens, divide them into 2 equal groups, and then further 4 groups, and we will provide them with the waters that we got in objective 1. And then we will challenge, we will vaccinate them against the IBV. We will challenge these birds with the IBV virulent strain. And we will be looking at the clinical signs, virus shedding in oropharyngeal and cloacal swab, and the virus load in respiratory, GIT, reproductive, and renal tract. We will also be doing histopathology on the effect of virus in various tissues, and also immunohistochemistry to detect the viral antigen in the tissue. At different time points in this control experiment, we will also be collecting serums and doing antibody titers of the vaccine and the challenge. We will also be doing cell-mediated immune response measurement. We will take monocytes from lungs and spleen and stimulate them with a virus lysate, and take the supernatant for interferon gamma production and measure with the interferon gamma ELISA. We will also go for CD4 and CD8 specific interferon gamma production by intracellular staining, and finally measuring it with flow cytometry. This was all about my project. I would like to thank all committee members of my research, Dr. Faisal Karim, who is the PI of this project, Sylvia Checkley, Dr. Dong Yang Niu, and Dr. Susan Kaur. And I also like to acknowledge my lab team members and all the funders of this, the Egg Farms of Alberta, Higher Education Commission of Pakistan, who is funding my PhD, University of Calgary, NSERC, RADR, and Canadian Poultry Research Council. Thank you for your attention, questions, and comments.
Thank you for another great presentation. That sounds like a relatively ambitious project with all the analysis, in vitro analysis to be done. And I'm looking forward to hear more about the results in a couple of years, hopefully.
Yeah, sure, welcome.
So I've got a couple of questions here, and I just remind everyone to use the Q&A box to submit your questions. From myself here, I was wondering about the vaccination program. When you're going to be sampling all those farms, are all the vaccination programs and the type of vaccine used similar across the farm, or is that going to be taken into consideration in the analysis?
Yeah, that's a great question. The vaccine that normally there is a routine uniform vaccination. I mean about the strategy of vaccination performed by the Alberta Layer farmers. They use live vaccination initially, and then they boost it with kill vaccination. I'm like, just take example of IBV. That is the strategy they follow. And one of the reasons that we are doing multiple time point samplings, 18-day, 18 week, 30 week, and 45 weeks, that is to just see that various vaccine has effect and in titers at different time points, different titers. So we just want to see just that we are not confused on only one time points, we are taking multiple time points.
Okay. Yeah, and relative to the vaccine, there I see there's a question posted. Some producers use avian encephalomyelitis vaccine as well, and there's a suggestion it may be worthwhile to include that in your sampling.
Yeah, that is a great suggestion. So that's a kind of vaccine, yeah, not performed by all the farmers, because the 3 vaccines that we selected is kind of uniform vaccination strategy done by almost every farmer in egg layer in Alberta. The encephalomyelitis, it's a kind of off and on vaccination strategy. We can think over it if if we have complete data from the farm, if every farmer or most of the farmers that we are going to sample are doing this vaccine strategy, we can include it in the sampling.
Okay, well, thank you very much. And here one more questions on the question box. What is your criteria for labeling a water a bad quality water? Is it chemical, or is it physical, or biological, or all of them? So I assume the question is, what is the criteria to be labeled bad water?
Yeah, I understand that. That is a tricky one. So ideally, our criteria will be all the 3. Like if a water is bad in physical, chemical, as well as microbial characteristic, that is an ideal bad water. And apart from this, if we don't get this, then we can include 2 of them. If not only one, the microbiological contamination can make our water bad. So that's, we don't have a threshold that only that every water falling compelling all the 3 characteristics is a bad water. We can variate it, but at least we are also going to compare this water with the tape water, the municipal water. So there must be difference. That's our comparison, or the control is the tape water. So there must be difference. We will evaluate all the 3 characteristics of water quality.
Yeah, and thank you for that answer. That kind of also answers the next questions that we have here. How are you going to find different water with different quality? Will the water source be different? Will you manipulate water to create different categories?
Yeah, we are now going to manipulate. That is, we will be using the same water that is that's provided to the birds on the farms, whatever the water is, because they are using those waters, and we analyze those water. So if a water comes in a good quality, then it will be categorized as a good quality. And if it is a bad quality, because the test shows the water is bad quality, so then we will be further using that water for our controlled experiments. We're not going to manipulate it.
All right, thank you very much for answering all those questions. And